Good morning, everyone. Our first speaker is Ms. Carolyn M. She will be speaking on extended reality applications in urology. A bit about Carolyn. She is a fourth-year medical student out of Johns Hopkins. She attended Duke University for undergrad. She studied biology and documentary studies. And outside of urology, Carolyn is interested in medical education and humanities and medicine. So let's give a warm welcome to Ms. Carolyn.
Hi, everyone. Again, my name's Carolyn M. It's great to be here visiting for the month. I've really enjoyed exploring New Haven so far. And I'll be presenting today on extended reality application within urology. I don't have any disclosures. And in terms of an outline of what I'll be covering today, I'm going to go into a bit of background of what extended reality is and some definitions on that, and then talk about some studies that have explored extended reality applications in diagnostics, preoperative planning, intraoperative guidance, patient education, as well as medical education. So what exactly is extended reality, or XR? It's actually an umbrella term that encompasses virtual reality, augmented reality, and mixed reality. And so starting from left to right, working away across the screen, virtual reality is when you have a digital setting that's completely isolated from the real world. Augmented reality is when you have digital objects that are superimposed onto a real world view. And then mixed reality is when you have real and virtual objects that are intertwined and can interact with each other in the environment. And when we think about extended reality, this isn't necessarily something that's far and near off in the future. There are a lot of commercially available devices on the market right now, including Apple's Vision, Microsoft has the HoloLens, Magic has Leap, and then Meta has the Quest device out. And in pricing, there's a huge range of what's available on the market, from $500 to $5,000 of the device that you end up picking. But I think the more interesting point is that the number of expected users is predicted to reach nearly 4 million people by 2028. So it's a technology that, is expected to become more ubiquitous amongst the population. And so first off, I wanted to start off exploring how urology and diagnostics could use this technology. And it's been explored as a way to look at the accuracy and efficiency of prostate biopsies. And so Zogel and colleagues ultimately ended up taking preoperative imaging, using real-time ultrasounds, and then fusing that imaging to the real-time ultrasounds. So you're able to have an overlaid image and navigate in real time as you're doing the biopsies. With this setup, you could also have a trajectory that is plotted out in order to see where your actual biopsy would be placed. And ultimately what they found is a success rate defined as cancer detection from the first biopsy of 70% versus just 30% of just 2D ultrasounds. And this isn't necessarily something that's new. It's been implemented and used widely. In terms of preoperative planning, another study has looked at using standard MRI imaging versus 3D virtual reality versus 3D printed models in terms of prostatectomy planning. And so what this looked at is taking 10 patients who retrospectively had biopsy positive lesions, and then asking radiologists to look at the imaging, identify out the lesions, and then segment out the anatomy to inform the creation of both a 3D printed model and a 3D virtual reality model. From here, urologists could then take one of these modalities, and they were asked to identify lesions on a PIRADS map, and then this could be graded for accuracy against the radiologists to see how accurate the urologists were depending on the modality they were given. Additionally, the urologists were also given surveys in terms of how easy the modality or model was to use for them, and if it helped in terms of their confidence in lesion identification. And so here you can see example models of what the urologist would be looking at. On the left is the virtual reality model, right, 3D printed model, and you can see you're able to manipulate it in space. The segmented anatomy is color coded. And in the case of the virtual reality model, you're able to remove portions and see the interior structures as well. Ultimately, what they ended up finding was that in comparison to just standard imaging, that the 3D models were more helpful in terms of accuracy of lesion identification, the speed and confidence and ease in interpretation from the urologist standpoint. If you take a click down and look specifically at the 3D models, so this would be the 3D printed models or the 3D virtual reality models, ultimately 3D printed models were found to be the most helpful. And so I think this is an important point of thinking about not only how we can apply extended reality in certain settings, but also being thoughtful in doing comparison studies to see if it's actually the most helpful technology that you could use. In terms of preoperative planning, it's not just been limited to prostatectomies. There have also been studies exploring the use of these technologies within kidney tumors. And so for the first study, Wellens and colleagues looked at Wilms tumors in pediatric patients. And what they did was they used, again, 3D printing or augmented reality to see if surgeons rated visualization and understanding of the tumor to be more helpful. And it was noted that with the models that the surgeons did have improved anatomical understanding. And then in terms of partial nephrectomies, Yoshida and colleagues have also explored the use of mixed reality. It's an example here of the digital image overlaid into your real world environment. And the idea would be that preoperatively, they actually use an Xbox controller, which I think is kind of funny, to manipulate the model in space. And then if you were in the actual OR, in order to remain sterile, you'd be able to use your hands freely in space in order to manipulate the model and move it in within a 3D fashion. And they also noted that the surgeons felt that it provided increased understanding in terms of anatomy of the tumor. So the next portion I'd like to introduce is an application called IRIS. And so I'm going to take you through the flow of how IRIS might be incorporated, and then we'll look at some studies of the efficacy of potentially incorporating this technology. So if you had a patient who came in and they had preoperative imaging done, potentially what you could do is get that preoperative imaging uploaded into this iOS application called Iris. You can actually download it onto your smartphone device. And then with this, you'd be given a 3D anatomical model that you could manipulate on your smartphone device. And then also it could be linked with DaVinci to be seen intraoperatively. And so this is an example of intraoperatively of what you might be able to see. So you can see the 3D anatomical model at the bottom that you could have seen preoperatively via your smartphone, and you can use it while you're in real time looking at structures in order to cross-check and compare from the actual DaVinci robot. So I think the natural next question is, okay, great that you can do this, but does this actually potentially have an impact on the surgeries that are being done? And so this study looked at, if we're using IRIS, can you actually improve surgical precision of partial nephrectomies? And what they ended up doing was they took preoperative imaging and were able to find the volume of the kidney preoperatively, and then predict out with the volume of the tumor removed what the postoperative volume of your kidney minus your tumor would be. From here, they could then take postoperative imaging, calculate the volume of the actual kidney, and then compare the predicted versus actual whole kidney volumes after surgeries. And what they found was that for patients who underwent IRIS-guided procedures, that they actually had significantly improved precision in terms of that difference between predicted and actual kidney volumes. I will say, that being said, this didn't necessarily translate to clinical significance because their 3-month out kidney function values weren't necessarily statistically significant, but an interesting thought that potentially as this improves, it could be used to improve the way that the precision is conducted intraoperatively. So, intraoperatively, there have also been studies exploring the use of augmented reality in terms of detecting capsular involvements in radical prostatectomies. And so, for this particular study, they ended up taking preoperative imaging and then fusing that with intraoperative views, as you can see here. And so, you were able to see the overlying structures and then tag them with where you think there might be capsular involvement based on imaging. And so with this, they ended up taking a metallic clip and at the nerve sparing phase of the surgery, tagging that portion of where they thought that capsular involvement might be. And then once pathology came back, comparing that and seeing if it was actually indeed involved. And in the cases where you did have augmented reality reconstructions, this overlaid image, they really were successful in identifying capsular involvement within these surgeries as compared to just standard nerve sparing without the augmented reality overlay. Some of the drawbacks to this, however, are you did have to have someone manually come and link the two images, so it was time intensive. But what's cool about this is if you have the overlaid image, they were able to also get it to be able to be elastically deformed, meaning that your overlaid image in terms of navigation wouldn't necessarily stay static as you're moving around dynamically within the human's body. So there have also been studies looking at partial nephrectomy outcomes. This was a retrospective study that was done comparing 3D augmented reality partial nephrectomies versus using just ultrasound imaging in patients with renal tumors. And what they found was that you were able to have a higher concordance when you had augmented reality in terms of your intra-op management of your renal pedicle as opposed to preoperative planning. They also found that they had higher rates of enucleation, lower rates of global ischemia, lower rates of collecting system violation, and then at a 3 month out checkpoint, reduced impact to your renal plasma flow. This being said, this was, retrospective, so I'd be interested to see if they do more controlled studies coming out, but interesting to think about potentially applying these technologies to improve the way that surgeries are conducted intraoperatively and potentially improving patient outcomes afterwards. So next, moving to patient education, there have also been studies looking at how these models can be used to help patients better understand their disease and their treatment plans. And so in this case, they took 200 patients undergoing a variety of urology related procedures, and then they randomized them to receive education either by standard imaging or imaging plus a 3D model that was patient specific. And these 3D models were either printed, they were augmented reality, or they were 3D models that were viewed on a 2D screen. And ultimately, what they found was that 3D printed models were rated by patients to be the most helpful in terms of them understanding their disease, their cancer size, location, treatment plan, and their overall comfort level with the procedure moving forward. So I think there's this idea that if you have a model or you have some of these things, it doesn't necessarily have to be isolated or siloed to one part of the patient journey. You could potentially use it preoperatively for planning, intraoperatively for guidance, but also perioperatively with the patient to help them be more comfortable with what's happening and going forward. And the patients noted that, even though 3D printed models were found to be the most helpful, that actually any of the 3D models any of the 3D education tools that were available were rated to be useful for them. So potentially, identifying one and taking the time with your patient could help them ultimately be happier and more comfortable with what's going forward. And then in terms of medical education, this technology has also been explored as a way to help more junior trainees become comfortable with different procedures. At a medical student level, they've explored the use of mixed reality for training digital rectal exams. And so this image shows an example of a model that a learner is standing at, and then an overlaid instructor hand that is able to help guide and provide feedback to the learner. Additionally, I think what's interesting is you're able to also bring up the internal organs on the view of the learner so that as they're feeling the model, they can also visually see internally what they're actually feeling for. And so this was rated to be useful by trainees and potentially could be helpful as especially more junior learners are learning things where it's difficult to visualize the internal anatomy. Additionally, at the resident level, they've also explored the use of extended reality in terms of IPP placements. And in a residency cohort who did learning with IPPs, they were found that the vast majority really liked the learning that happened and wanted it incorporated in their residency program. And then finally, what I'll speak a little bit more about is the use of mixed reality for potentially teaching residents how to perform biopsies. And the end kind of takeaway spoiler is that mixed reality was equally as effective as in-person learning in learning this procedure. And so for this setup, what they did is Dr. Ghazi, who's actually now on faculty at Hopkins, took hydrogel simulation models that were equipped with giving ways of giving quantitative feedback to the learners in terms of how well they were performing their biopsies. You have your smart glasses in the bottom left that would allow the learner to see the overlaid instructor hands, and then mixed reality software where you're able to have the instructor remotely give gesturing feedback, verbal feedback to your learner while allowing them to remain having control and having autonomy over their learning methods. And this would be an example of the setup that you would see. So on the main screen, you have your instructor remotely able to give instructions to your learner. On the bottom left, your learner setup is there using smart glasses and also in front of a model. And then on your bottom right is the view that both people are able to see. And again, you can see as the instructor gestures, you're able to overlay their hand into the view that the learner is seeing. So I think one of the things residents really liked about this feedback wise was that while the learner is getting instruction, both verbally again and through gesturing, hands-on, they are ultimately still able to have full control over, the equipment they're using and really get a grasp on how to perform the biopsy. As I mentioned, the mixed reality was equally as effective to in-person learning in terms of learning the biopsy procedure. That being said, the majority of residents actually did prefer in-person still, citing issues like technical difficulties and timeliness feedback issues. But I do think as this is like a newer technology, as it becomes more ubiquitous, as I mentioned in the beginning of the presentation, and some of these kinks are worked out, that there's still a lot of potential, especially in areas of where urology teaching in person might not be accessible, to expand outreach from that perspective. So some overall limitations on some of the studies I've spoken about, though it was in that one case with capsular detection, it's not always that if you have, extended reality overlays that they are dynamic. And so if you have static renderings, it doesn't always necessarily translate to what's happening intraoperatively. In some of the studies we talked about, extended reality actually wasn't the best possible thing that was found to be used. And so I think being really thoughtful about when to apply it and studying in comparison, not just in silos of the technology out there, it's important. A lot of the studies also, were retrospective in nature. They had small sample sizes. And at this point, because the technology is newer, it is still expensive and you do have to put upfront learning time as well as setting up the equipment and linking the imaging in order to use them. So some overall final thoughts. We've gone through some applications within urology that extended reality has been explored in. In other surgical fields, it's also definitely something that's being explored, but I think it's really exciting to potentially see as more people are adopting the technology of where it could go. That being said, I think it's still really important to be thoughtful and to have evidence-based medicine being practiced. And so there's more research that needs to be done in terms of where this could actually be used to improve patient outcomes. And as it becomes more popular, I think these devices will only become more accessible. The costs will come down. People will understand better how to use it. And then finally, something I'm particularly excited about because I like medical education is, expanding outreach in places where you don't necessarily have urology educators to help more people learn remotely is something that is really interesting.
Our next speaker is one of our sub-Is visiting from the University of California, San Francisco, Mr. Than Kyaw. Today's talk is entitled Harnessing the Microbiome to Advance Urologic Science. Than is an MD PhD student, as I stated, visited from the University of California, San Francisco. His passion for the microbial ecology was ignited during an expedition to Mariana Trench while pursuing his undergraduate studies at UC San Diego. At UCSF, his research focuses on the human microbiome as a National Cancer Institute fellow. He's interested in understanding the influence of microbiome on urologic diseases and treatment outcomes. Let's give a warm welcome, Mr. Dan.
Hi, everyone. I'm Fan. I'm from UCSF. I'm really excited to be here and talking to you all about my passion for the microbiome research and application for the urological sciences. So today we'll talk about how microbes are expert chemists that live in our body, who can change the chemotherapeutic efficacy and toxicity. We'll also talk about incorporation of the microbiome into human genetics, host genetics, and disease characteristics to prognosticate and predict the outcomes in designing personalized medicine. And the third point is that microbiome is an underutilized tool right now to really understand the pathogenesis and treatment outcome in urology. So over the last few years, there's been a lot of studies that shows the microbiome is linked to various urologic diseases like stones, infertility, prostatitis, cancer like prostate, bladder cancer, chemotherapy efficacy, and neobladders and augmentations in reconstructive urology. So for the purposes of my talk today, I'm going to focus on two topics, the chemotherapy and ureteroplasty. So let's start with the chemotherapy first. The capecitabine, which is a chemotherapeutics, is an oral equivalent of 5-fluorouracil, is used to treat metastatic breast cancer and colorectal cancers, but people are now trying to see if it could be applied to treat muscle invasive bladder cancer. The problem with capecitabine, though, is that it's highly variable. FDA calls it a highly variable drugs because more than 50% of the patients require dose modifications. We can't predict how they're going to respond. It's such a wild drug, but it works. So we ask, can the gut microbiome change the CAP efficacy and help explain the variations in treatment response? So the way CAP works is it is a prodrug. It gets activated into 5-fluorouracil and then deactivated through this enzyme called DPYD. We found a human equivalent in bacteria called the pre-TA enzyme that does the exact same chemical reaction. So we wonder, does the pre-TA and bacterial enzyme reduce treatment efficacy? So we designed this causality experiment using mouse tumor models where we injected the mice with human colorectal cancer cells. And then we colonized them with E. coli that has no pre-TA or E. coli that has a lot of pre-TA. And the reason why we're using E. coli is because I can manipulate his genome super quick. Like I can knock out his genome overnight. So we split the mice into two groups. We either give them no treatment, the vehicle, or treated with capecitabine. What we predict is that in the vehicle groups, the tumors will grow really big because they're not getting treatment. In these CAP groups, because there is no pre-TA to interfere the way CAPs work, the CAP will work as magic and tumors will be really small. But however, in this group, capecitabine is being degraded or inactivated by pre-TA, so it wouldn't do its magic and tumors would keep growing. That's our hypothesis, and here's the actual results. So as predicted, the vehicle groups have big tumors in both pre-TA knockout groups with no pre-TA or in group with a lot of pre-TA. In here, without pre-TA, capecitabine works and it shrinks the tumors. But in this group, the tumors kept growing. And the only difference between these two groups is the amount of bacterial enzyme. Everything else is the same. They're getting the same amount of treatment, the same tumor dose, and we kept every other parameters the same. The only thing we manipulate is the difference in the microbial load. They even got the same microbes. So that really shows that microbiome significantly alters the chemotherapeutic efficacy, and this experiment shows the evidence of causality, not just an association. And the implications, which I'm really excited by, is that, right now we use host genetics and disease characteristics to really predict and design personalized medicine for our patients. But what this study shows is that we really should be thinking about how the microbiome is playing a significant role in terms of the treatment outcomes. So in the future, I think we'll be able to incorporate microbiome as part of the equation to predict and prognosticate the disease outcomes and the treatment outcomes. And this study is a proof of concept study to look at the microbiome interactions, and that could be applied to other urologic diseases. For example, we can find a bacteria, find the gene, develop an animal model, and then test the pharmacokinetics and pharmacodynamics in urologic drugs. So we published this paper in Nature Microbiology, and then we did this clinical study that's currently being resubmitted to Science Translational Medicine. So hopefully that'll be out sometimes in the near future. At this point, I was really excited to apply my skills in the microbiome science into the field of urology. And one of the studies that we did that really shows the translational nature of the skills into urology is the project I did with Dr. Ben Breyer and Dr. Lindsay Hampson in terms of looking at the urinary microbiome for urethroplasty project. As we know, the buccal urethroplasty is a very well tolerated cornerstone treatment for urethral strictures, but the complication still remains, like stricture recurrence. What's really exciting about this procedure is that during this procedure, when you're grafting the buccal mucosa, you're not only taking the buccal tissue, but also the microbiome with it. Because you're not sterilizing that piece of tissue, you're just, engrafting it and putting it into the urinary tract. So we asked, what happens to these oral microbes now that it's in a very different environment in this urinary tract? So what does the microbiome look like post-urethroplasty? And does the persistence of the oral microbes play a role in the surgical outcomes and the complication rate? So what we did was a pilot study where we had 11 people. One of them didn't need a buccal graft during the operations, and we collected the intraoperative baseline samples where we just collected urines during the procedure. And then we looked at the post-operative samples that were collected at 3 months of clinical visits. Unfortunately, one of them got incarcerated, so that's automatic rule out since he becomes ineligible. And then 3 people didn't show up to their 3-month post-op appointment, so we weren't able to collect it. So our baseline samples had 9 people, and then our post-operative samples had 5 people. And then we basically looked at the buccal, the mouth microbiome, and the urinary microbiome pre and post procedure. And here is a 30,000-foot view of what the microbiome looks like. And the way to interpret this graph is each sample is portrayed as a dot. The warm colors, the orange ones are the oral swabs, and the green ones are the urine. And circle is the baseline that we collected intraoperatively, and then the final ones are the triangle. The patients are connected by these dotted lines. And the way to interpret this graph is that the more similar the microbiome are, they'll appear closer together. So as you can see, the oral samples all clustered together and the urine samples all clustered together, meaning oral and urine microbiome profiles are very distinct. But what's really exciting about is that in the oral swabs, there's not very much changes in the baseline and the final time points. They're all kind of clustered together. But in the urine, you can see huge changes pre and post procedure, meaning the microbiome has changed somehow. And you might wonder what changes are happening in the urinary tract. So here's another graph. This is a volcano plot where on the x-axis is the full change, looking at the final versus compared to the baseline. So the positive numbers means higher in the final, negative numbers means higher in the baseline. And on the y-axis is the significance of the p-value. So in the urine, we see that there is a high enrichment of Corynebacterium after the buccal urethroplasty. And Corynebacterium is a mouth-associated microbe. It's been implicated in gingivitis. It's been implicated in dental abscesses. So what is it doing in the urinary tract, like 3 months after this buccal urethroplasty procedure, and what could be impacting the host? So that's super exciting to me, and we're really excited to explore this. So the buccal urethroplasty was associated with changes in the urinary microbiota and with the enrichment of Corynebacteria, which is the mouth-associated microbes. And the implication is that this is a new way or a tool to really explore microbial ecology, how microbes communicate to each other and how they form communities, because they're not living by themselves. They are living in members with other microbes, and they're constantly communicating and talking with us. And buccal urethroplasty, being a reconstructive urologist, you guys have such a new way to look at how microbes form. Because what we're doing is you're taking the buccal tissue and combining with the urinary tissues to create this chimeric environment that doesn't exist in nature. So you're creating a piece of land and you get to watch it develop as a urologist, right? So is it going to be like rich and rich rainforest that doesn't have strictures and is in healthy environment, or is it going to develop into such like a barren desert full of strictures? And who are the founding microbes that really determine this trajectory of the surgical outcomes? So this is, I'm really excited to use this tool to really probe how microbial ecology forms and what are the principles of microbial ecology. You might be wondering how the microbiome research might fit into the really busy urology residency. So what I've been thinking is that microbiome research is inherently collaborative and translational with high payload, because it's easier to publish right now, and it has a very high impact because people are very excited about it, and it's inherently translational because we're constantly going from bench to bedside and bedside to bench. And it is feasible during to do research during residency, in my opinion, and I haven't done residency, so this is just all postulation, hypothesis. But the microbiome sampling is non-invasive and is easy to add into other studies. So in terms of like getting IRB approvals and things like that, we never really had any issues. They were like, if you want urine, go for it. Like if you want stool samples, like I don't really have a problem with this. So it's much easier to get an IRB approval, and in terms of like getting through all the red tapes. And analysis is mostly computational. So we don't have to be in the lab and doing a lot of lab work. Once the data, once the sequencing is done, which is mostly now outsourced to companies, we have lots of sequencing data, and you can kind of sit through on your computers and do all of these analyses. So I think that, that really assists in terms of doing research during residency. And what I will focus on is the microbiome impact on urologic oncology and reconstructive or pediatric urology. So the two projects that I've been thinking about in terms of applying these skills and doing projects in residency is the urologic oncology project. One proposal that I've been thinking about is that how the BCG impacts the microbiome. So BCG, as we know, is a attenuated form of tuberculosis bacterium and is a standard treatment for non-invasive, non-muscle invasive bladder cancer. But about 30 to 40% of people don't respond to this treatment. And why is that? Well, BCG relies on the immune system to work, right? Because it activates the immune system. We know that the microbiome trains the immune system. It is a huge educational center for immune system to develop. We have these tire patches that lines the intestines that trains the immune system to recognize who are friends and who are foes. They're constantly sampling the intestinal lumen to really figure out who are the microbes that are present and should we mount an immune response. What if that training center is wrong? Right? Like what if there's an aberrant microbiome that really trains the immune system to recognize something different? That would inherently impact the way the BCG works, because BCG relies on the immune system to work correctly. So perhaps variations in the microbiome may alter the immune response, impacting the BCG treatment. So what we would look at is, let's take a look at BCG responders and non-responders. Let's take a look at how the microbiome is different and the immune profiles are different. And then go into the lab and develop a cell model to figure out how the microbes, how the immune system responds in the presence of in the presence of microbiome from the BCG responders versus non-responders. And we can translate that into a xenobiotic mouse model. Another project is that I've been thinking about is the augmented bladders. We know the augmented bladders are constantly colonized because there's the piece of intestines that supports the high microbial payload colonizations in their native system. But 30 to 60% of people get symptomatic UTIs. Not everybody gets them. So why do some get symptomatic UTIs versus some don't? Perhaps differences in the microbial communities may account for the symptomatic UTI development. What we will look at is again, taking a look at how the differences in the bladder microbiomes of individuals with and without the symptomatic UTIs in the longitudinal study. So we'll just collect those samples and sequence what the microbial community looks like and what the urinary metabolome looks like. What are they doing to assess their activity? And I think Yale is a good place to do the microbiome work because you guys have a really rich microbiome community. For example, the chair of microbial pathogenesis, Dr. Andrew Goodman, just recently published, his trainee just recently published in Nature that shows that the gut microbiome carcinogen metabolism causes bladder cancer. What they did was they fed the microbes nitrosamine, which is the carcinogens, into mice, and then they looked at differences in the microbiome in terms of mice that developed urinary bladder cancer versus who don't. So that's really exciting. And you guys have such a diverse staff and people who are experts in different fields. So it's such a collaborative environment to do healthy, strong microbiome work. So really excited to share this talk with you all. And here are my acknowledgements. I really like to thank my PI, Peter Turnbow, where I did my PhD work. Here are my collaborators, my urology research mentor, Dr. Cobb, Dr. Hansen, Dr. Breyer, Dr. Shaw, and I'm funded by the F30 National Cancer Institute and T32 for my medical scientist training program.